Hi everybody, Miss Chotzi here. I wanted to go over the question that was posted in Google Classroom as a sample question of something you would ask to complete on a test or a quiz. So what was given to you was a sequence of DNA. I can identify this as DNA because of the presence of thymine, so I know that it is in fact a sequence of DNA. The first job that you need to do, or the real goal, is to figure out what is the amino acid sequence that we would make from this particular DNA. So what we need to do is transcribe it into messenger RNA. So this process of turning DNA into messenger RNA is called transcription. And transcription, by the way, happens inside the nucleus because that is where the DNA is. And because the DNA cannot leave the nucleus, RNA's job is to get the message out of the nucleus to the ribosome where you can then build the protein, because a protein is made up of a sequence of amino acids. So when you want to transcribe DNA into mRNA, you do base pairing rules, except remember that in RNA there's going to be no thymine. So you won't write any T's in this line. You would replace it with U. So T would pair with A would now pair with U, and C would pair with G. So there's our first one. Then we have UCA. Then we have U. Oh, I thought I made a mistake there. It's really easy to make mistakes here. UGG. Then we have CGA. And then we have GUA. And finally we have UAA. P.S. Each one of these is called a codon. Okay, so a codon is a sequence of 3 bases on a messenger RNA, and that sequence is then going to correspond to a particular amino acid. So our last step here is to build our amino acid chain. Sometimes this is called a polypeptide. Peptide bonds are what hold amino acids together. And I would be building this not in the nucleus now, but now I would be constructing my amino acid chain, which will eventually become a protein inside the ribosome. So what you do is you use your codon table. So that's really what this was. This is a table of all the possible codons. And by the way, there's 64 combinations of A, U, G, and C together in triplet form, but there are only 20 amino acids. So you can clearly see that a lot of them are used more than once. Now, how and why in terms of evolution did these codes pertain to these things? That is a mystery that I do not have an answer to, and I'm not really sure if anybody does. So what you would do is search for AUG on your table. Here it is right here. It codes for the amino acid methionine, which is always, by the way, the first amino acid in every gene. This is called our start codon. Sometimes they'll actually just write start, depending on who made the table. Next one we would find is UCA. You search for it on your table and you come up with something called serine, which is another amino acid. Then we find UGG, that is TRP, which is tryptophan. Tryptophan, P.S., is found in high doses in poultry, and when humans consume lots of tryptophan in their diet, they get very sleepy. So that's probably why people get that Thanksgiving coma where you get very fatigued after you eat a lot of turkey, because there's a lot of tryptophan in turkey. Random thought. Okay, next one is CGA. This is going to be arginine, ARG. Next we have GUA, which is valine, VAL. And then lastly, UAA is one of our stop codons, of which there are 3 of them up here in red. So every gene is going to end in one of the 3. Okay, so that would be the sequence of amino acids that you would build from the sample question. One thing that I didn't mention, which I want to mention because it's going to be on a further task I'm going to have you do a little bit later, is what is the role of tRNA in this in this business. What is going on? What does tRNA do? Remember, it's the transfer molecule. So basically, how do each one of these guys or amino acids get to the ribosomes? They are hitched on, they are attached to a tRNA molecule. So the corresponding tRNA would be the complementary of the mRNA. So the tRNA code would read UAC. And this is called an anticodon. Okay, so these are anticodons. So the tRNA molecule, remember, is kind of a T shape. And at the bottom of the T are these like 3 really important bases. And at the top of the T is going to be the amino acid. So that is a terrible drawing. There are much better ones online and in your book, but that's kind of the idea. So this will match up with our mRNA, and then we can assemble and build our protein together. So the next tRNA would be AGU. The next one would be complementary to UGG would be ACC. Then we have GCU. Then we have, man, this is hard. CAU. And then finally, we have AUU. I hope I didn't make any mistakes there. So remember that each one of these amino acids is attached to a particular anticodon or a particular tRNA molecule. So this is the anticodon. So they really serve no other purpose than the transfer or the movement of these molecules to the ribosome. So when the right one collides with AUG, then you can build this together. So they're all in the cytoplasm, colliding and present. It's just that when the right two find each other, they'll form a bond, and then you'll start to build your sequence of amino acids. And then the bond that holds these together is called the peptide bond, okay? And that's how you make a polypeptide. Now, in reality, a real gene would be way longer than this. Way longer than this. This is a very short sequence. Okay, hope that helps, and there'll be more practice soon.